All right, this is the DNA lectures. Okay, on the screen right now, you're looking at a DNA double helix, right? And notice that the DNA double helix wraps itself around some protein beads. We call these protein beads histones. This would be the first time you hear about them. But the wrapping around them sort of helps organize and keep the DNA threads from getting all balled up and kind of turn into a big mess. I'm sure you've had experience with like string and things like that where if you ball it up, it's kind of difficult to unravel it back into a string. So something similar would happen at the molecular level with our DNA double helix if we didn't have these histones to help. The histones are also important for a variety of other reasons. Those histones can be chemically altered in such a way that they actually ball up the DNA in a way where it's not accessible. And once we learned about that, we could turn off genes by balling up or really super tightening the DNA by chemically altering the histones. So we could turn off genes that way. We can also make genes a little bit more accessible by also chemically altering those histones. So they're kind of important, is what I'm trying to say. They're not just important in organizing the DNA, but they're also important in determining whether you have access to a gene or not, just depending on how tightly you coil the DNA and the histones around each other. Notice that the histones that have the DNA coiled around them also then begin to coil around themselves here. And then that coils even more into bigger and bigger things. And then eventually we come to the recognizable chromosome, right? That we've been discussing in mitosis and meiosis and in genetics. That is part of the law of segregation, part of the law of independent assortment. We saw crossing over in prophase 1. We separate sister chromatids, so that's one sister chromatid, that's another one. That's the centromere that connects the two together, right? Okay, so what we're going to do in this lecture is we're going to get down to the details of how did the DNA become duplicated during interphase of mitosis and meiosis. But to get there, we're going to have to do a little bit of history. All right, on this slide, I want to take you back to the early 1920s when a lot of people were dying of bacterial infections, something that today is more of a rare occurrence, right? Our medical fields have advanced to the degree that we can, for the most part, easily treat bacterial infections. And we also know more about sanitation and hygiene and stuff like that, so we reduce the likelihoods of bacterial infections, right? So the combination of all of that has made bacteria less of an issue. We might have a more significant issue with viruses and things like cancers and all that, so our medical fields are now a lot more focused maybe on some of that, although they still dispense antibiotics and so on. All right, so let's go back to the early 1920s where bacterial infections were a real big issue, right? And in this case, we're going to talk about a bacterial strain called pneumococcal pneumoniae. So it's a bacteria that causes pneumonia, as the term implies. And it came in two forms. One form is described as being rough, and the other one is described as being smooth. The rough form is called rough because when it grows on a petri dish, right, those petri dishes that you used in lab this week, those petri dishes would have bacteria growing on it, and the bacteria would look a little bit textured. So we described it as rough. But the other strain grew on those same petri dishes, and it looked smooth, glossy. So we called it the smooth strain. Well, in the early studies of this bacteria that was known to cause pneumonia that people would die of, we took the bacteria in its two forms, and in the rough strain, we injected it into these mice, and then waited to see what would happen to them, and you can see that they all lived, they all survived. And if we euthanized them and looked into their blood, we would find that the mice didn't have any evidence of bacteria, even though we had injected them with the bacteria. And so what that suggested is that their immune systems were strong enough to fight off the bacteria. When we tried it with the smooth strain and injected that into the mice, all the mice died. And when we looked at their blood, we found that it was just loaded with the smooth strain. And that suggested that the immune system of the mice was not strong enough to fight off the smooth strain. Later, we determined that the reason why they were smooth is because they had a capsule. And you might remember from a long time ago when we first discussed bacteria that have capsules around their surface, right, around their cell walls, they're able to withstand the attack of our white blood cells. So they're medically very relevant to us, right? In the medical field, we worry about these kinds of bacteria that produce capsules because they can protect themselves from our white blood cells. And clearly here, that capsule protected them so much that they ended up killing the mice. Okay, so then in the third experiment, so that was experiment 1, 2. In the third experiment, we took the bacteria that was smooth and then we boiled it for a very long time and created basically heat-killed bacteria. So they're dead. I think you understand that we denatured all their proteins, and so we destroyed them. We injected that soup into the mice, and it looks like the mice survived. So clearly the only way to kill the mice with with the smooth strain is to give them living smooth strain. But if we give them some of the soup that contains parts of what used to be alive, that doesn't kill them. All right, in the fourth and final experiment, this is the one that I want to focus your attention on. In this case, we inject 2 non-deadly things into the mice. We injected, again, the rough strain that does not kill, and we injected the soup of the heat-killed smooth strain that also doesn't kill. So we put them together here, and then we inject it into the mice, and as you can see, all the mice die. But if that's surprising, what's even more surprising is that when we look in the blood of the mice, we don't find the rough strain. That's gone. And instead we find bunches of smooth strain. So what happened? Well, back then we knew that, life would not have spontaneously shown up. So we figured that the rough strain turned into smooth strain. And we called that, back in the day, a transformation. And I'm hoping you'll remember that that was the topic of lab this week, right? We talked about a transformation. This is when one bacteria with a chromosome and a bunch of little plasmids potentially ruptures open, it lyses, releasing all of its little plasmids, and then another bacteria in the surrounding area absorbs the plasmid. So that's what happened here. The rough strain that was heat killed and turned into a soup contained a bunch of little plasmids. And when we mixed that liquid with the rough strain took in the plasmids, and now we're able to produce a capsule around themselves that protected them from white blood cells. So they became genetically transformed. It turns out that in nature, genetic modification happens naturally. Nature creates GMOs. All right. So that experiment was conducted in the 1920s by Frederick Griffith, and he called the transformation cause a transformation principle. He didn't know what exactly caused the transformation. So that created a race to try and figure it out. And so back then we knew about the biomolecules, right? Carbs, lipids, proteins, and something called a nucleic acid that was found inside the nucleus and was acidic. So we did a bunch of experiments, and I want to fast forward past all of those, right? And we made a discovery of a virus called a bacteriophage. So this is a virus that is very specific to bacteria. And when the virus lands on a bacteria, it drops its DNA into the bacteria. The DNA is read by the bacteria. It uses the instructions to make a bunch of viral proteins and also replicates the DNA. And then the viral particles begin to assemble themselves. That's part of the developmental stages of a virus. And then all the viruses, once fully assembled, they all come out at the same time, causing lysis, right? Complete destruction of the bacteria. And so a bacteriophage is literally a bacteria eater. A phage is something that eats. So we call it a bacteria eater because when we released the virus onto bacteria, it seemed to dissolve them away like it was eating them. All right, this is what the bacteriophage looks like. So I want you to record that term, bacteriophage, and know what that is. It's a virus that infects specifically bacteria. And this is a kind of a really cool cartoon model of that virus, but the actual virus looks very much like this. And we know it from electron micrograph images. And so what it does is it lands on the surface of a bacteria, and then it uses some sort of like hypodermic needle that you can see here in the background, and it uses that to snake in its DNA into the bacteria, and then the bacteria will use that DNA and create more viruses, and that's part of the viral life cycle, just like that, right? So any one of these viruses can then go back and do it again to another cell. Okay, so what we learned from this experiment is that the virus is made actually of two things. It's made out of protein, and it also has DNA. And it looks like the protein stays outside, and the only thing that goes in is the DNA. And then all of a sudden, the bacteria changes what it used to do. It wasn't making viruses, and now it is. Before the viral infection, it wasn't making viruses, and now it is. So something changed its genotype, which in turn affected its phenotype. We changed its genes by adding more DNA, and as a result, its phenotype changed. It changed from not creating viruses to all of a sudden creating viruses. And what this helped us determine was that protein, which was doing everything back in the day, and we thought was the agent of heredity and such, wasn't actually the cause of the transformation. It was actually DNA. And so now we knew that DNA was the root cause of heredity, genetics, and in this case, explained how viruses replicate, and also helped us figure out what caused the transformation. It was DNA all along, but the very first molecule we considered heavily was protein because it seemed to do so much other stuff. It just made sense. All right, so for those of you moving on to microbiology, transformations are an important concept to know about. Whenever bacteria can absorb DNA from the surrounding environment, that's a transformation. But bacteria can also exchange genetic information with each other by viral infections, so that would be phages, and that's called transduction, right? So when viruses are part of the exchange of DNA, that's called transduction. You're going to learn this in microbiology, so we're not going to make a big deal out of this for our course. We're going to stick to transformation. It's part of our lectures. It's also part of our lab, so we'll definitely focus on that. A third way in which bacteria can exchange genetic information is through a process called conjugation, and this is where one bacteria with a bunch of little plasmids parks next to another bacteria that has its own chromosome and maybe also some plasmids, and then they create a tube between each other, and they make a copy of a plasmid, and then they pass it over, and they can exchange plasmids that way. That's called conjugation. All right, so there's 3 different ways in which bacteria can transfer DNA within the same generation, like no one else can do. when we pass our DNA on to others, we pass it on to the following generation, not to the same generation. But bacteria are able to do that. All right, let's talk about DNA now a little bit more closely now that we know it's a little bit of its history and how we discovered its role in life. What you're looking at is rope ladder. Yeah, that's rope ladder. And you can see that if you twist it, it'll look more like the DNA double helix. So that's just to kind of get your mind prepared for looking at something a little bit more complicated like this. So here's a DNA double helix in a kind of simplified cartoon form. And over here is the form that I want you to really think about. This is another form that we use, the ball and stick model, to show the spatial arrangement of all the components of DNA. But let's stick to that middle one, right? I want you to first notice what the DNA seems to be made out of. It's made out of a repeating unit, a subunit, repeated over and over again. And it's also repeated here, here, and so on. There's a total of 8 of those units here, and all of them are called nucleotides. So all DNA is made, in fact, all nucleic acids, DNA and RNA, they're all made of nucleotides. So that's what these are. Now the nucleotides themselves are made up of 3 parts. They all are made out of a phosphate. Right, you can see the phosphate here. They're all made out of a 5-carbon sugar. We call it a pentose for 5 carbons. That's this guy here. And finally, they're made out of a nitrogenous base. And you can see they all have that base right there. So those are the 3 parts of a nucleotide. It doesn't matter if we're talking about DNA, which is this case that we're looking at, or if we're talking about RNA. RNA is also made up of nucleotides, and those nucleotides are made out of the same 3 parts. We're going to tease out the differences between DNA and RNA a little bit later. All right, what I also want to point out is that, let me try and clean this up a little bit. I want to point out that the DNA strands also have some patterns within them. For example, the DNA strand on the left seems to be going in one direction, whereas the one on the right seems to be going in an opposite direction. And we call that anti-parallel. I'll write it up here. The 2 strands are anti, meaning opposite directions, and still running parallel to each other. Oops. Whoa. So they're parallel to each other. Another thing to point out is a pattern of bonding. You can see the hydrogen bond here that is holding one strand to the other strand. Whenever there's an A, it's always attached to a T, and whenever you see a G, it's always attached to a C, and it never changes. That's a very important pattern to recognize. A's with T's, G's with C's, always, always, always. We call this the complementary base pairing rules. Complementary means one complements the other. So what complements T, it would be A. What complements C, it would be G, and that never changes. Those base pairings, they always happen that way, and so they become rules. All right, so those are 2 important things to get from there. Notice that they all have the same exact phosphate. Notice that they all have the same exact sugar. But the only difference is the base. So let's look at the bases now. All right, here's all the bases of DNA plus one base found in RNA. So let's focus our attention here. Notice that thymine, abbreviated T, and cytosine, abbreviated C, they are lumped together into a group called the pyrimidines. And that's because they are each made up of one ring. This is an organic chemistry term. They're called pyrimidines. On the other hand, look at adenine, abbreviated A, and guanine, abbreviated G. Those are lumped into a group called the purines, and that's because they're made up of two rings. Right? 1, 2 rings. So again, that's an organic chemistry term. So you want to know what group belongs to the pyrimidines and which ones belong to the purines. So these are these A's and T's that you were seeing in the previous slide. That was adenine with thymine, guanine with cytosine. You were looking at these. There is a fifth base, and that one is called uracil. And uracil, notice, is made up of one ring, so it belongs to which group? The pyrimidines or the purines? Belongs to the pyrimidines. It's one ring, just like thymine and cytosine. Okay, let's move on. Let's look at a very broad way, a cartoon, simplified way of DNA replication. This is the parent DNA. This is basically the DNA double helix. And we're about to replicate that DNA double helix. And you can see that the first thing we do is we separate the strands. So if I were to maybe do this, you're seeing that we separated the strands. That strand and that strand have been separated like these have been separated. And then what we do is we copy each strand. But notice the way we copy them. If you see an A, you'd put down a T. So it's not identical copying, it's complementary copying. If you see a C, you put down a G. If you see a G, you put down a C. All right, same over here. If you see a T, you put down an A. You see a G, you put down a C. Uh-oh, what's going on here? That shouldn't go there, right? That's going to be a mistake. But sometimes that happens, and that's called a mutation. That's how mutations happen. That's one way mutations happen anyway. Okay, but let's say that we do everything correctly, like demonstrated here and here. If we do everything correctly, then we will make a copy of that guy and a copy of that guy, and then they'll begin to condense themselves really tightly, and then that's how we will have produced. Oh, I'm going to have to stretch way over there. This is how we produce the chromosome. And a chromosome is made up of two identical what? Sister chromatids. That's what we made there. But these would be stuck to each other. Those two would be stuck to each other, and they would form a chromosome. But individually, they are sister chromatids. So these two right here are actually these two right there. All right, that's DNA replication. Let's dig a little further, because I think you understand complementary base pairing. I think you understand having to separate DNA strands so that you can have access to the parent DNA so that you can make copies of each one. And I think that you get that the copies are not identical, they're complementary. But if you do both of them the same exact way, do you see that this one is identical to that one? And we know that sister chromatids are identical. All right, so let's see it a little bit more complicated. There's your DNA double helix right there. Here's the separation of the parent DNA. And then in gray, you can see the copies being made, and they're all complementary. And at the end, you should notice that this guy is identical to that guy. Those are the sister chromatids. Okay, let's talk DNA orientation, because that's super important. All right, here's that DNA double helix again, largely simplified. 4 nucleotides on the left, 4 nucleotides on the right. But now there's numbers associated with them. Those numbers are important. All right, here's the way we come up with the numbers. The sugar is, again, made up of 5 carbons. Those 5 carbons are the source of the numbering. The first carbon is always wherever the base is attached. Here's the base. It's attached to the sugar there, so we call that the 1 carbon. Or, I'm sorry, the 1 carbon. And then from there, we just go clockwise. And we go 2, 3, 4, and 5th carbon. The 5th carbon is always attached to the phosphate. So I think that's potentially a mnemonic you can use. A mnemonic is a way to remember things. 5, phosphate. 5th carbon, phosphate. Okay. If you wanted to attach another nucleotide to that one right there, you would have the nearest carbon be the number 5. So we call this the 5' for that reason. Because that carbon is sticking out with a phosphate for something else to attach to it. So we call it the 5'. If we were to apply the same logic back down here, that's the 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5th carbon is right there. Do you see that if you wanted to attach something down below, you would have to interact with this alcohol, which is attached to which carbon? The 3. So we call this the 3', and that's where the numbers come from. And remember that this strand goes this way and this one goes down below. They're anti-parallel. So if this is the 5', then this must be the 3'. If this is the 3', then this must be the 5'. Okay, now we've created orientation, and now we can start getting closer to replicating DNA. All right, so one more time, here's your DNA double helix. Here's a very simplified way of showing just one DNA strand where we don't highlight the sugars or the phosphates. We're mostly concerned with the bases A, C, T, G, and G. And we can show you the nucleotides a little bit better here. There's the A nucleotide, C, T, G, and G. And you can also see a pattern of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So that's called the sugar-phosphate backbone. But that's going to be identical with all DNA. It never changes. The thing that matters to us are the bases. Whatever order the bases are in, that's what's different between you and I and your neighbor and your parents and other animals, other species, plants included. It's the order of those bases that matters. All right, remember again, all DNA, all RNA are made up of nucleotides. The DNA nucleotide is made up of 3 parts: phosphate, sugar, base. Okay, let's get ready to copy DNA. And then I'll show a video on this presentation. Okay, let's start over here on this side of the slide. Notice that when we simplify the DNA to just be like 2 strands attached to each other, that's what is represented here in teal. Notice that when we start copying DNA, we can start here, here, and there. And when we start there, we call it origins of replication. So that's an origin of replication, that's an origin of replication, that's an origin of replication. And they start out by forming bubbles. That whole thing is a bubble. That's a bubble. That's a bubble. And notice that the bubbles are expanding outward and eventually converge with each other. And when they do, you will have created 2 sister chromatids, here described as daughter DNA molecules, but you know them to be 2 sister chromatids. The point of that is that when we copy DNA, we don't start at one end and go all the way to the opposite end. We start in multiple locations in between, and then we work our way out until we converge with bubbles. And then we complete copying the DNA super fast. And for us, super fast is still like, 6-7 hours. Okay, what if I take one of these bubbles and I chop it in half, right? Then I would have a fork. I would have this. There's the fork, highlighted in dark. And notice that now we're adding numbers, the 3 and the 5s that we saw in the previous slide. Here. And that's important because when we copy DNA, we copy it by reading it always from 3 towards 5. And when we make a new copy, the new copy has to be from 5 to 3. All right, we're going to get into those details now. How do we copy DNA? So I already have a slide prepped for that. I'm going to give you some rules, and I'm going to give you some enzymes. First, the rules for copying DNA. And for the exam, all you have to know are the rules and the enzymes. There are 3 rules that you want to know. 1, you need an RNA primer to get started. You'll see what this means in a second. Think primers as in paint. Let's make that RNA in red. Second rule. We always read DNA. Always, always, always. 3' towards 5'. Rule number 3. The new DNA copy is always 5' to 3'. Those are the rules. The enzymes that we need will be 3. Number 1, well, let's just stop with that. We need DNA helicase. This is an enzyme that unzips the DNA. See how the strands are separated on the right-hand side? That was done by DNA helicase. So this one breaks the hydrogen bonds that hold the DNA strands together. It breaks these bonds. So DNA helicase just cuts right down through there. All right, second enzyme we need is called DNA polymerase, as in a polymer. This is an enzyme that reads DNA 3 to 5 and also copies it. And it copies it 5 to 3. So, in other words, DNA polymerase is the reason for rules number 2 and 3 above. The last enzyme we need is called DNA ligase, as in liga, as in ligature. It's a binding enzyme. It binds or connects something called Okazaki fragments. Okay, we have our rules, we have our enzymes, and you want to know them. They're going to be on exam 3. Like, what are the rules for copying DNA? What does this enzyme do? Be prepared for that. Okay, first thing we need is a primer. There it is. That's the RNA primer. And you need the RNA primer because you need something to attach the new DNA strand to. I'm going to make my DNA strand, the new one, in blue. And so all I'm going to do is I'm going to read the DNA and I'm going to copy it. So if there were like a, for example, let's go back. If this was like A, T, C, C, G, T, C, A, what is the new copy going to be? It's going to be T, A, G, G, C, A, G, T. And they're connected to the RNA primer. The RNA primer is made up of RNA. All right, so we are reading the DNA from 3 towards 5, and we're making a new DNA strand that starts out 5, and over here it's at the 3 end. And it should look identical to the bottom strand. All right, here's what's interesting. It turns out that the bottom strand is also read from right to left, just like this one, the top strand. So we're going to read the bottom strand in the same direction. And you should recognize there's issues there. The rules say you have to read the DNA 3 to 5. So if I go from right to left, the bottom strand is 5 to 3. The top strand's fine, it's 3 to 5, but the bottom strand is 5 to 3. That's an issue. Well, we couldn't figure out how that was happening. We understood the rules, but we didn't understand how the bottom strand was being copied the same exact time as the top strand. This is where a video is going to help, but I'm going to explain what we learned. The problem was solved by a husband and wife team. They solved it. Their last names were Okazaki. And so they discovered something called Okazaki fragments. It turns out that the bottom strand is read in sections. There's a primer attached there, and then we extend. We'll have a combination of RNA and DNA. And then that repeats again. There's be a primer there, and then we extend. But notice that now we're reading from 3 to 5, but we're doing it in sections, and that's because the bottom strand gets flipped. See how it's now 5 there? But let's say I pick this area arbitrarily, it's 3. So basically what our cells do is they put a little primer there, and then they copy it 3 to 5. That's how these were made. And then that just continues over and over again. We can put another primer there, another primer, let's say here, and we would have been making copies of it. Always the same way, 3 to 5, 3 to 5, 3 to 5. So once we have all those fragments, notice that there's a lot of primers in between them. That's kind of a big deal. We now have to get rid of those primers, and we have to replace them with actual DNA. And when we do that, then we have to connect the fragments together, and so that's the job of DNA ligase. It connects, it binds together the Okazaki fragments. Okay, so let's show that. I'm going to take a eraser and I'm going to erase all the primers. Oh, overdid that one. Oh, and overdid that one too. Let's put back that DNA strand. What? All right, there's the DNA strand back, and then let's put back some of the primers that we had here. All right, I'm sorry, the DNA fragments. And now we need an enzyme to fill in what's missing where the primers used to be. But then we also need an enzyme that comes in and connects them. Connect them, right? Connect them. Connect the fragments of DNA, connect those two fragments together. Let's show a video of that.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, we'll stop the video or the lecture here. We've just covered DNA replication. In the next video, I'm going to pick up with a process called transcription and translation. How do we turn DNA into RNA, and how do we read RNA with ribosomes to create protein?